afternoon, everyone. I'm Dr. Charu Yadav from Medanta, the Medicity, Gurugram. So, having heard about the role of mass spectrometry both in clinical research and diagnostics, let us move on to hear the viewpoints of our experts in the field of healthcare. So, to begin with, my first question to Dr. Seema Bhargava is, we have seen that use in clinical research is there, use in diagnostic is there, and of course, mass spectrometry is a very precise technology. So what is there, your viewpoint on that bridging the gap, what all we can do in clinical mass spectrometry from clinical research to diagnostics continuum? So ma'am, before we begin, can you put on some light on the how was mass spectrometry discovered or invented in the first place?
Thank you, Charu. And I'd like to always go to the history of anything because it is humbling to know that such a beautiful technology was developed from one person observing one small thing long ago. And that long ago was not very long ago. It was in 1898 when the German scientist Wilhelm Wien, he observed that a beam of charged particles, when put in when exposed to an electrical field, but deflected. And that was the first basis that was set by him for mass spectrometry. And then, as Dr. Sunita has already told you, that in 1900, the British physicist J.J. Thomson, who had already discovered the electron, found that the electron, and he observed that its deflection by an electrical field was there, and he then conducted an experiment in which he applied electrical as well as magnetic fields in different directions, which today we know as electromagnetic fields, but then it was not known at all. And he found that the deflection was in two beams which were perpendicular to each other, and if you put a photographic plate at the end of it, they formed parabolas. And these parabolas indicated that each parabola was of one specific was of particles that were of one specific mass to charge ratio. And its position on the parabola was according to its velocity. So yet one step further. And going another step further, we had, in 20 years later, we had Aston, who was a British scientist, and Dempster, who was a American scientist. And they individually formed spectroscopes where serial electromagnetic fields were applied to beams. And these actually deflected them in these particles in such a way that they were focused. Any particle of a single mass was focused to one point, and that is what we have today as mass spectrometry. That was the basic, very basic. And what we need to know is that the journey is very long even after that. And as Dr. Sunita has said, that it came into clinical use only in the 80s, but actually this was all happening in the field of physics. And its transition to biology came when, I forget the names of these, when Alfred Neer exponented its migration from the tight realm of physics to biology when they were doing traces studies on C13 enriched carbon. So that is the basis of spectroscopy, how spectroscopy came into being.
Thank you so much, ma'am, for bringing that insight. So my next question is to Dr. Anuj. Having known that it was initially just in physics and then translated onto biology, would you highlight some specific areas or diseases where mass spectrometry has shown exceptional promise as a diagnostic tool, and how can we further harness its potential?
Thank you, Dr. Charu. Good afternoon, everyone. So I think before I start, I think just before this panel discussion, we had two talks where Dr. Rao and Dr. Sunita has mentioned very about two particular conditions, which is one is the cancer, where he talked about the metabolomics. It is something which is coming up. I mean, of course, it's there since few years, but in cancer diagnostic area, the metabolomics is one such, or proteomics is one such area where it has a long application. And Dr. Rao actually has already given us how the kits have been developed by various companies like Thermo Fisher is one such who is offering this as a diagnostic tool. Psych is another such company which gives these tools. So these other companies, I mean, they're coming up with a kind of a mix of the cancer detection at the level of metabolic derangements. They're able to capture the metabolic derangement at the level of each analyte, each at the level of enzymes. So that is the one basic, or I would say a very, very revolutionary tool in the hands of cancer diagnostics. Another tool which is being used very, very commonly by pediatricians is, of course, the inborn error of metabolism. Dr. Sunita has already told us how it is being used and how the children are being saved in time, because we know that the time is essence in diagnosing children, especially who had these deficiencies of enzymes or metabolic disorders, what we call them. And this is actually being used in our hospital as well. Of course, we also getting such queries from our pediatricians, and we are getting this tested done. Apart from these two major areas, there's one area which is called therapeutic drug monitoring. We know in the area of transplants, the drug monitoring is in essence to salvage or to kill any transplant. So drugs like tacrolimus, cyclosporins, methotrexates we do have multiple technologies to detect these drugs, but of course the mass spectrometry is one technology which is sort of give you very accurate values of these drugs. So you can use this technology in this area of TDMs. Another is the area of vitamins and hormones. We know we can detect and even trace elements, for example, zinc, boron. So all of these elements which are important in all our metabolic reactions. So this can be used in these detection of trace minerals, metals, vitamins and hormones. It has also found to be role in the case of neurological disorders. It has been found that it has a role to identify special proteins in case of Alzheimer's disease. In fact, I just came to know that infectious disease also has, it can detect even those peptides which are there in infectious disorders. So it has a multiple applications, just to list few. But yes, the first and foremost, the two major applications which has been the, I would say the best possible use of this technology. One is the metabolomics for cancer detection, and another is inborn error of metabolism for pediatrics or neurology. Thank you.
Thank you, sir, for giving us the broad prospects. Moving on to Dr. Shaloo. Ma'am, would you share your views on what are the scope and business opportunities dealing with this advanced technology that is mass spec?
Good afternoon, everyone. There is wide scope and there are wide applications of mass spec. I think so. I'm audible to everyone. Yeah, there is wide scope and wide application of mass spectrometry. And as Dr. Anuj and Dr. Rao and all have explained that, and Dr. Sunita also, that the first and foremost, the basic use is basically in metabolomics. That is in analytical chemistry. Basically, it is used mainly in the newborn screening, as well as in hemoglobin analysis, as well as in drug testing. That is therapeutic drug monitoring. That is the main role of mass spectrometry. Going ahead, we can use it in pharmacs, that is in pharmaceutical companies are using it for drug discovery, be it drug pharmacokinetics, as well as in the drug discovery is one key area where mass spectrometry has major role. Another thing is that in environmental studies, it has been found to be very much effective, be it in the water quality testing, food industry, food safety. This is one area where mass spectrometry has major role in the industry. Then in the also in the geological studies, what it has been found that in the oil composition as well as in the hydrocarbon estimation in the petroleum industry, mass spectrometry has major role. In metrology, estimation of the trace elements as little as parts per quadrillion, mass spectrometry has found to be very effective. Now, in the dope testing of the athletes, mass spectrometer is the most effective methodology basically. Also in the biotechnology, for the estimation of the peptides, as well as in the other, as Dr. Anuj also has told, and all our other panelists have also discussed, that peptide analysis also, mass spectrometry has major role. Now with the omics technology being used more and more in this era, and its application being highlighted, mass spectrometry is one of the key areas which is going to grow, and with proteomics being the driving force, mass spectrometer is one basic methodology which will be helpful in discovery of all these biomarkers which are there in the upcoming time, and we are going to see that its use will be very much highlighted in the medical industry, and this is my opinion about it.
Thank you, ma'am, for your inputs. My next question is to Dr. Seema, ma'am.
And one more thing, sorry,
Yes. No.
As far as the industry is concerned, at present the industry is having around 4 to 4.5 billion, and it is with a CAGR of 7.5%. It is estimated to grow up till 11 billion by the year 2030-31. So the growth is quite a lot. The market size is going to increase a lot, and we are going to find that with the mass spectrometer being used so much, that we should think, I think so, it has a future. So we should see that we all should be open to use it. And the clinicians are definitely very much fascinated. The only issue is that user friendliness, as I will discuss in the upcoming points.
Thank you, ma'am, for bringing out that scope as well. So, Dr. Seema, ma'am, so since you told that it has been invented decades ago, why has this methodology become more important and gained important in the recent years?
This technology is very, very specific, but it takes time. It has taken a long time to develop because it was first not in the realm of biology. And once it came into the realm of biology, those things happen. But what is equally important also is that methodologies for simple molecular testing, I don't mean molecular as in molecular biology, but I just mean for testing of analytes and for testing of molecules is available by other methods also. And therefore, the focus was on those. At that time, our knowledge about the human physiology and biochemistry was restricted to a liver function test or a renal function test. And these tests were very well done by different methodologies, by spectrophotometry, and as these methods evolved, they became faster and faster, and they became more sensitive and more specific also. And therefore, the clinical needs were taken care of. However, as medicine advanced into the molecular biology phase, where genetics, epigenetics, metabolomics, all of these have evolved, the very high sensitivity of this particular methodology, where it measures each and every atom or molecule of the substance that you're asking for, that becomes very important. And what we have is, you may see that in the future. Today we talk about it being very useful only in these smaller molecules and in the metabolomics. But you never know in the future if in the clinical scenario, smaller and smaller differences in analytes will be correlating with different morbidities and clinical presentations and different therapeutics. We might even shift all those. And by then, I'm sure even mass spectrometry will be on a very different, fast platform. Today, it is not very fast, but it might be in the future. Never say never.
Thank you so much, ma'am, for very rightly pointing out the issue. My next question is to Dr. Anuj. So, since ma'am told there have been multiple methods for the general analytes, which are the most commonly used tests, how can we promote collaboration between researchers, clinicians, and industry stakeholders to advance the clinical utility of mass spectrometry?
I think as Dr. Seema has just mentioned that it was in the realm of physicists and it came to biology. Of course, it took some time to come into biology arena. And then, so now basically, again, the question is that how do we collab? So there are three stakeholders here, the researchers, the clinicians, and the industry. Now we have to bring all these three stakeholders on a common platform. And I think the best way could be, I think the way today we are doing this discussion here, I think this is the first thing to start with. You should have a lot of conversations, conferences, workshops to bring all these three entities together on a platform. I think that's what Thermo Fisher has done while he bring three of us or of a hospital-based, industry-based researchers on a same platform. So that's the beginning, or I would say the start in this journey. Second, it has to be a very collaborative research projects. We have to have a lot of research sort of activities with academias, with industry has to support the hospitals and the academias to bring this projects to, let's say, to our hospital-based reams. Networking and platforms, we can have, as I said, this is an area where we can have a lot of network. Thermo Fisher or eHealth or can make this journey to bring all of us together and to have this ball rolling. I think the moment we'll start speaking on this topic more, we may find more avenues, more applications for this technology. We should be open in our data sharing and databases. The one major challenge is that people do not kind of share their databases, and this is something I have seen. So until unless we share our databases, until unless we share our data with each other, with that openness, that would bring more, I would say, momentum again in this journey. So I think we can have, again, the help of Thermo Fisher to brought in this part. The next thing is one thing I think we were discussing yesterday, the training and education. This is something is lacking in this area. I think I have read it when I was doing my MD about this technology, and after that, it just went off the radar. So we want that we should have more trainings and workshops on this technology. It should be made available to at UG level, PG level, and after that at hospital-based levels. Industry collaboration is very, very required. We really can't, run this race or this journey alone unless we have a help or support of industry leaders. And we have multiple industry leaders in this area. So I would urge all of the industry which is present here to help in collaborations with the hospitals. And of course, the funding is one area where we always find short of. If we have them some sort of opportunities to get that grants or funds from non-profit organizations or from industry, that would help us to bridge this gap. So these are my thoughts on this particular topic.
Thank you, sir. Very rightly said that we need more collaboration in both awareness and knowledge learning based research. Moving on to Dr. Shaloo. Ma'am, same question to you. What do you think, in your viewpoint, are the drivers for growth in the field of mass spectrometry?
No, actually, the driving force is investment. Again, investment in R&D as it has been done in the pharmaceuticals. Now, this, as Dr. Seema has already highlighted, that identification of target molecules, that helps in the growth of therapeutic medicines. Like it helps in the identification of all these targeted therapies. Now, this all happens through mass spectrometer. Now, you identify all these targeted molecules through mass spectrometer. So, the growth of these awareness about these, the information about these targeted molecules can only be generated through these mass spectrometers. So, this requires investment, and investment in R&D is one key area which is going to be the driving force for growth of this industry. Now, growth of mass spectrometer. Another key point is, as the first one important thing is the segment which has the maximum usage of mass spectrometer is Pharmac. Pharmac has the mass spectrometer maximum usage because of its using for drug discovery. Similarly, the segment which is going to have the maximum usage in the coming years is life sciences, and for that we have to again have the investment from the both for the R&D, both for the omics and proteomics for the usage for proteomics for the discovery of biomolecules as well as the target molecules, all these biomarkers especially. For these, of course, again the investment in R&D is required. Another thing, an increased usage of mass spectrometer requires is better performance on the part of mass spectrometers, those mass spectrometers which are more user friendly, which have better accuracy, and all these things, of course, is there. And again, the final and most important point is again collaboration, as already highlighted by our co-panelists. Because collaboration amongst all the key stakeholders, be it the researchers, be it the clinicians, be it the diagnosticians, and of course, like Thermo Fisher and other companies, they can all work together to make such type of mass spectrometers which are more user friendly, those mass spectrometers which are handled well by the scientists, and which are able to generate data, and which are able to which have more throughput, and those which are faster, which are able to have a better TAT. Those all things can, of course, in the years to come, have better be the driving force for increase of these mass spectrometers be usage in the industry in the years to come. And definitely one important thing is that the mass spectrometers, the clinicians are very much fascinated by the mass spectrometers because the technology is definitely fascinating, as we all must be aware. But at the end users are the one who are facing the challenges, and definitely we have to understand them, and then we can proceed.
Thank you so much, ma'am. So next question is to Dr. Seema. How can we promote appropriate use of this method as well as the newer techniques in the future?
Thank you again. And first of all, we have to understand that in a clinical scenario, in a routine laboratory, it may not be a very, very popular thing because what the doctor is dealing with is mostly emergency medicine. If you're talking about emergency medicine, you're talking about something that you need a result before you've given the sample, which is typically how they call us. Before we receive the sample, we get a call from the clinician. What is the result? So in that scenario, with these tight expected turnaround times, mass spectrometry as it stands today cannot be used for routine analytes. But the world of metabolomics, genetics, epigenetics, toxicology, as Dr. Shaloo has also very rightly, and Dr. Anuj has also pointed out about therapeutic drug monitoring, about antifolates and anti-immunosuppressants. So in all these fields, we are going to find a change, maybe even a shift from the routine from the routine platforms to the mass spectrometry, except that at the present moment, it may not be cost effective. But we all know that what was not cost effective yesterday is cost effective today. And the way technology is increasing, we find that even in mass spectrometry, in many areas, they are already trying to make automated platforms. And automated platforms, and the other very great advantage about mass spectrometry is that they have, these are certain panels available to you, 1,000 metabolomes or whatever, metabolites and these many molecules. But beyond that, you can also develop something. So what I feel is that in future, we need to start very early. We need to teach our children and teach our students that what is the basis of all this? How did it come up? Open their minds, make them think out of the box, make them look at how can one small reaction be converted or maybe developed into a methodology or maybe a therapeutic method. And for that, there are many things like, and physics and biology, they have to go hand in glove. Not just physics, chemistry, biology, physiology, all of them have to go together because they are the ones. For example, Bhabha Atomic Research Center has a technology transfer group. And they were the ones who brought irradiation as gamma rays as a method of irradiation of sterilization of medical instruments. And that was brought to the medical field. It may not be very popular, there are many places where it is used, but that was the first time, and I know that only because my father was the director at that time of the technology group. So what we need to do is we need everyone to work together, to keep thinking. We have to encourage people to think out of the box. Please do not just go by textbooks.
Thank you so much, ma'am. Very rightly said that we need more trainings in the field. So maybe the seniors in their respective fields can promote such learning experience among inter-institution collaborations. Moving on to Dr. Anuj. Sir, in a country like India, what do you think the economic considerations such as instrument cost are playing an important role when it comes to widespread usage of mass spectrometry in clinical settings? So, what do you think is the role of economic considerations and how can we address such potential barriers?
Of course, you said that right. A country like ours, we always have economic considerations is our biggest question mark, which comes in everybody's mind. And coming from a lab industry point of view, whenever we put this proposal, just to give you my real life example on this particular technology, I was in RGCI before Medanta, and that time, it's a cancer hospital. And I put this proposal to my director to get mass spectrometry for metabolomics, I think 2-3 years back. So he told, you call that person. We called that person. He came. And the moment he gave us the cost of that instrument, he said, he looked at me, my face, my boss, and I looked at his face. And then we both had this kind of understanding that it's not for us. So obviously the economic consideration would be the most important point while getting in the hospital-based setups. So this is something we have to discuss in the form of an initial capital investment. We have to talk in the terms of ROIs. We have to see how much is the cost incurring versus how much is your returns. So that factor obviously has to be discussed, has to be seen, has to be understood by administrators. And now, how can we facilitate this particular cost, which is the starting point for this technology, and which I'm saying it's huge. And consider coming from a lab side where we always have this sort of reagent rental models, where we do not put our money in the huge amount of money in the investment in the first hand. So maybe the industry has to come up solutions or a ways that how to help the labs or the hospitals with some sort of either from third-party financing or some sort of rental models. So they have to find a way, or maybe we all have to find a way how to get around this economic considerations. Then, of course, one thing is your test costs rationalization. You have to see how much your volumes you are generating and how much it is feasible to have that test in-house. Currently, for example, we are outsourcing this test to our network lab partners. And for them, it makes sense to do this test because it's a test which needs volumes. And unless we have these volumes, again, so that's another point. We have to see, can we make a nodal centers where we, like, for example, one hospital take a lead that all inborn error of metabolism will come to us and all metabolomics comes to another center. So some sort of arrangement has to be made or seen that way. And of course, the cost of maintenance, consumables, all have to be factored in while going ahead with this instrument or technology.
Thank you, sir. Very rightly summarized all the implementations. So maybe the industry support system can find out ways apart from just buying the instrument. Maybe there are ways where we all can implement this in our respective setups. Moving on to Dr. Shaloo. Ma'am, having talked about the opportunities of mass spectrometry in clinical labs, could you share your experience in what challenges or limiting factors have you faced in implementation of this system? And what are the recent advances in mass spectrometry?
See, the limiting factors, as already few of them have been enumerated by my co-panelists. Actually, one of it is the throughput. All these mass spectrometers over the years, they have to have a higher throughput. And more important is that they have to have the shorter testing times, and the faster the test is performed then definitely it will be more acceptable, because the turnaround time is definitely the key for all. Means it's one of the key indicators in all the labs. So that is one important thing. Another thing is that we have to have a skilled personnel, because be it not only the instruments handling, even the data handling, both ways. So both hardware as well as the software, pairing of both have to be handled well, and that way, if the data processing, and already one important thing is there, that the data processing and all the data should be available to everybody. This is one thing is there regarding mass spectrometer. The clarity of data is also one thing that is required, and that's a limiting factor for mass spectrometer. Another thing is that few of the mass spectrometers even need to be portable also. Actually, that is also one of the issues regarding mass spectrometer, because the era is there of the that I will talk about the future aspects. Cost, as already discussed, cost is one key thing. They have to be cost effective. And then more important is that there are even challenges even in the inborn error of metabolism also. That is, at times the mass spectrometers, they are giving actually they have this, this is one of the technical aspects, that those with the same mass, few of the, all biochemists can highlight this very well, that like leucine and isoleucine and all, they will all be highlighted as similar. So that is also one of the key challenges. So for that you require further chromatography, you have to be advised and all those things. So coupling of chromatography as well as the mass spectrometry, all these things means it has to be improved. The technology has to be, the technological advancements have to be done in the mass spectrometer to make it more accurate, so that the mass spectrum, the spectrum is better, more clarity is there, more precision is there in the spectrum, so that it is more acceptable for and more clarity in the results are there over the period of time. And it is we are answering the clinicians that, see, this is the final, the final result is this, and over the years then it will be this. These are a few of the limiting factors with the mass spectrometer. Now, as far as the future aspects of mass spectrometer is concerned, of course the future is bright, but the way ahead is definitely, way forward is definitely bright. But definitely there are one thing is there is future is in proteomics. Like, I come from a cardiac setup and the clinicians are very much receptive regarding it. But then again, the proteomic analysis of the cardiac myocytes is the way ahead for the in mass spectrometer. Be it in the multiple myeloma, be it is one interesting thing about the mass spectrometer is that it is replacing IFE. That is, the immunofixation is also over the years it is going to replace, as it is able to differentiate the M protein from the therapeutic antibodies. So monitoring of the therapeutic antibodies is also one key area where the improvement of the mass spectrometer is going to grow, and the future is very bright regarding that also. Another thing is there is as already Dr. Anuj and Dr. Seema have said, that in the cancer profiling, mass spectrometer has lot of advancements is being done, and proteoforms of the prostate carcinoma, this is being developed, and definitely prostatic carcinoma. Of course, other biomarkers already are being highlighted, but the proteoforms of prostate carcinoma is being developed, and this is one key area where mass spectrometer is going to grow, and it has a great future. But overall, what I feel is that on all levels simultaneously, in all levels of omics, be it lipidomics, be it genomics, and be it proteomics, mass spectrometer can be run in all the three means, can have a key role in all, and definitely the future is bright.
Thank you so much, ma'am. Just before we end the panel discussion, just a quick question to Dr. Kanuri, if he is here. Dr. Kanuri is still present with us? Okay. All right. Dr. Sunita is here? Okay, no issue. Okay. Thank you so much, panelists, for rightly summarizing the challenges we have in this very much advanced technology. Although it is precise, it is the most sensitive way out, but still it is lacking implementation, at least in the hospital settings. Research, yes, it has gained some importance, but bringing it to hospital, actually patient care and diagnostics, is still a road ahead. So thank you all esteemed panelists for sharing your viewpoints.
Can I make one last comment?
Yes, please, ma'am.
I love the way Dr. Charu has made two, Shaloo. Dr. Shaloo has made two very variant statements. One was appreciating the sensitivity of it, that it is able to differentiate an M protein from the other IGs, the antibodies that are therapeutic antibodies. On the other hand, she says, well, it doesn't differentiate between isoleucine and leucine. So this is what our perspective is. It's increasing, it's changing over the years, and that is why mass spectrometry probably has a very great future.
Exactly.
Thank you so much, ma'am, for highlighting that very important point.